editing the so-called dark genome help crops survive extreme heat?
So heat tolerance is a very complex trait, and what people have tried doing before is like knowing one gene, like there's this family of heat shock proteins, and people would just put multiple copies of this inside. But a major part which no one has considered till now is like you put it in, it's basically switched on forever, right? Doesn't matter whether the stress is there or not.
Right.
And that has been the biggest problem for all these heat resilience genes that were out there. And that's what we're trying to solve by looking at the dark genome. So let me give you a fact. Only 2% of the genome is a coding region. So all these genes that we know of, that's only 2% of the whole genome of a plant. Around 98% of it is regions that are dark genome regions or regions that control this 2%, right? So of course this 2% is what is expressed, but the remaining part inside is what matters, because if you understand how that works, you basically control this 2% to make whatever new traits you want. And that's what we do at Plantik. We use a little bit of machine learning where we look into multi-omics data sets. So for example, genomics, transcriptomics, things that are linked to opening and closing of chromatin, which is known as ATAC-seq. So all these kind of multi-omics data sets, we look into that, and we have our own proprietary algorithm that then gives us the precise dark genome elements that can be fine-tuned to create new traits. For example, for heat resilience trait, we have come up with some of these switches which can be switched off, and because of that switching off, it gives you a better climate resilient tomato, for example. And this gene is only switched off when temperature goes up.
Right.
Rest of the time it's still functional. It still does whatever it needs to do as long as the temperature doesn't go up. And that's the major difference between what we are doing and what was the status quo till now in terms of breeding to create new plants.
I mean, how does your approach compare to, say, what Rainbow Crops is doing? They say they're using, AI, multiplex gene editing, and then high throughput phenotypic screening afterwards. Are you doing something kind of similar? Or?
No, we do something that is fundamentally different.
Yes.
By that I mean, again, most people are looking at these different gene regulatory networks.
Yes.
And they're trying to change some of these main factors,
Yes.
or like hub factors as they're called,
Yes.
which have multiple effects downstream, right?
Yeah.
You can play around with those, make some small changes there. But what we do is we are looking at those gene regulatory networks, but from a very different perspective.
Right.
We are looking at, so where do these transcription factors go and bind, and just change those few base pairs. At the end, the changes that we make are 6 to 8 base pairs, really small, and that has a much targeted effect compared to fine-tuning the whole network itself.
Yes.
And when that's where data sets come very important.
Yes.
That's why we need multimodal data sets.
Yes.
That's why a lot of data that we use is around ChIP-seq and protein binding microarrays, and that gives us an idea of for example, transcription factors that are activated under warm conditions. Where are they going and binding?
Yes.
And with this kind of knowledge, we are able to discover these exact sequences that then have an effect. And then we use machine learning for understanding all of this and applying it to plants where such kind of data sets do not exist, right? And that leads us to massively scale the platform to quickly discover new plants for which you do not have all the data sets available. So we basically use whatever is available, learn all these modalities from whatever is available, and apply that to crops where you do not have all the data sets available. We have most of our data sets built on tomato, corn, soybean, more modern major crops, right? And then we were looking at a data set in quinoa, and we were able to pinpoint mutations that lead to a quinoa plant being more tolerant towards salt stress.
Yes.
It has nothing to do with temperature, right?
No.
It's about salt stress.
Yes.
But the platform itself was able to pick these small nuances out of genes in the non-coding regions, or these dark genome elements, and it was able to tell us why some of these changes lead to a better salt-tolerant quinoa plant. And for that, we didn't have a lot of these multi-omics data set. We just had the sequencing data set, which today you can create for any new plant at a cost of $100, $200, And that's the bare minimum that's required to get started, to use our platform for discovering new traits. So our business model right now is mainly around being a tech platform.
Yes.
So we in-house we know what are our major focus traits for next 5-10 years,
Okay.
and we are working on creating those traits in-house, and then we will license them out to bigger seed companies to put them in the crops as
So
needed.
you'll license the actual traits, not
Traits.
the technology.
Yeah.
Yes. Okay.
So we would license traits out, but in future we see ourselves becoming more as a product company.
Yeah.
So for example, we have quite some interest from like wine growers, chocolate growers, berry growers.
Yeah.
So the idea would be to work with them to co-create new plants, which then goes onto the field, and we can collect revenues on products that end up in the market.
Yes.
So that's our long-term vision in how we would make money.
Yeah.
To help us reach that vision, initially we are doing small revenues from service projects, but also licensing some of our technology out to bigger seed companies.
Tell me about the regulatory kind of piece.
In US and in a bunch of other geographies around the world, these plants are considered as plants that are same that could be created through standard breeding. So that way it's not regulated as GMO. And even in the gene editing space, we fall in the category of NGT1, which is like New Genomic Technologies 1. And like these are again like where you have small minimal base pairs of less than 20 base pairs in the whole genome. And we are able to create new traits by doing precisely that, small edits of up to 10 base pairs to 20 base pairs.
Okay.
And that's why our plants currently, it takes around 1.5 months for every new plant that we create to get a regulatory approval in US. And then those plants can go straight to the field for growing and doing field testing. And last week, luckily, the European Union went ahead with the same regulations. They are going to build the same system as we have here in US. And in 2 years' time, we will have the same regulatory clearance timeline as we have here in the US. So 2 years from now, the new plants that we create, they would get regulatory approval in a time frame of a month or two. And that's going to be the case all around the world, India, Japan, and a bunch of other countries around the world.
Finally, what are the biggest challenges that you face as a business? Is it on the technical side? Is it about finding the right partners? Has it been raising money? You know, what keeps you awake at night?
Yeah, I think till now it has really been about funding. Like so, for this current round, we have been raising for last 16 months, I would say, and that's quite a long time. And every hour we spend on fundraising, we are not able to focus on building the technology itself, right? But I think all of that also had to do with a lot of regulatory red tape back in Europe, where we have our main operations, right? With that red tape opening up, I think funding-wise it's going to go faster, but confidence-wise, in having confidence on plants or traits that are created using new technologies, that is going to go up. And that we are already starting to see in terms of more inbound requests from bigger seed companies to try our platform out. For example, currently there's a big seed company from Netherlands which works on indoor tomatoes, and we already landed a project last year with them where they wanted to work on disease resistance for powdery mildew, and that's a project somewhere in tune of $250,000. And today we are having talks with them to build something similar for more traits around virus resistance, phytophthora resistance, and that would be closely to a tune of $1 million in terms of a service project. And this gives you the idea that as regulations kick in, open up the space for new players to join in. There's a lot of demand from existing companies to work together to create new plants. You know, at the end, like all these existing companies, they own most of the germline that are in market today, and it doesn't make sense for us to start all of that from scratch, but to stack our technology into existing germline that is currently marketed in the market, right?